Welcome to this video on drug development. We'll explore the rigorous process of testing and approving new medicines to ensure they are safe for patients. Why does drug development matter? Every medicine must be proven safe, effective, and stable. This process is essential to protect public health and prevent harm from ineffective or dangerous treatments. The two primary requirements for any new drug are safety and effectiveness. Safety means avoiding harmful side effects, while effectiveness ensures the drug actually treats the condition. Drug development follows clear, sequential stages. Each phase builds upon evidence gathered in the previous one, ensuring only the safest, most effective candidates move forward. We begin with preclinical testing. This is the first stage, conducted in labs and on animals to gather essential safety data before any humans are involved. Testing starts with cell cultures and tissue samples. This helps scientists observe drug interaction at a molecular level and rule out toxic candidates early on. Next, drugs are tested on animals, typically mice or rats. This allows researchers to study the effects on a whole, living organism systems before human exposure. During preclinical testing, scientists check three core areas: toxicity, appropriate dosage ranges, and whether the drug produces the intended biological activity. Toxicity is a critical safety check. Identifying harmful substances in the lab is more ethical and safer than discovering them during human trials. Establishing the correct dosage is key. We look for the therapeutic range, the balance where the drug is effective without becoming toxic or causing organ damage. Once a drug passes preclinical stages, it enters clinical trials. These controlled experiments involve human participants to further test safety and effectiveness. Phase 1 of clinical trials begins with healthy volunteers. This allows scientists to monitor safety in humans without the complications of pre-existing disease. Starting with healthy volunteers is an ethical safeguard. It isolates the drug's effects and protects vulnerable patients who are already unwell. In phases 2 and 3, the drug is tested on actual patients. These larger trials provide the critical evidence needed to confirm real-world benefits. Clinical trials always start with low doses that are gradually increased. This cautious approach ensures that any unexpected reactions can be identified early, prioritizing participant safety. Let's discuss the role of placebos. A placebo, an inactive treatment that looks exactly like the real drug. By using one, scientists create a control group to distinguish the drug's true effects from the psychological influence of being treated. The placebo effect is a well-documented phenomenon where patients report improvements in their symptoms simply because they believe they are receiving active treatment. This makes a placebo control group absolutely essential for reliable research. In double-blind trials, the defining feature is that neither the patient nor the doctor knows who is receiving the real drug. This concept is crucial for your GCSE exam. A double-blind trial progresses from recruiting participants and random assignment to the conduct of the trial, and finally, unblinding and analysis. The treatment codes are kept secret until all data is collected. Double-blind designs eliminate patient and doctor bias. By preventing expectations from influencing reported symptoms or results interpretation, they ensure the final data is scientifically valid and trustworthy. When comparing single-blind and double-blind trials for GCSE Biology, remember that double-blind is superior because it mitigates bias from both the patient and the researcher. Bias happens when results are influenced by factors other than the test variable, like hope or expectations. Double-blind trials remove these distortions, ensuring the data reflects the drug's true effect. Once a trial concludes, scientists perform a rigorous analysis. They compare the drug and placebo groups using statistical tests to confirm that any differences observed are meaningful, and not just due to chance. Meaningful results rely on three factors: a large sample size to reduce chance, statistical significance to confirm validity, and reproducibility, where consistent results are seen across different trials. For approval, drugs must prove safety and effectiveness. In the UK, agencies like the MHRA evaluate this evidence. Even after a drug is approved, long-term monitoring continues for rare side effects. The drug journey, from lab discovery through preclinical and clinical phases to regulatory approval, can span 10 to 15 years and cost billions. This reflects the immense care taken to protect patients. Why does it take so long? The process prioritizes safety, requires large sample sizes, includes long-term follow-up to detect delayed side effects, and involves rigorous, independent regulatory review. Avoid these common mistakes. Placebos are essential controls, not for deception. Double-blind means both the patient and the doctor are unaware. Also, preclinical testing includes cell and tissue studies, not just animals. For exam success, when asked why a double-blind trial is used, remember to state that both the patient and the doctor are blinded, which prevents bias and ensures reliability. Finally, here is a model answer. A placebo, which has no active drug, acts as a control. It helps scientists determine if improvements result from the medicine or the placebo effect. Let's review a sample exam question on preclinical testing. You might be asked to describe what happens. A model answer notes that the drug is tested on cells, tissues, and then animals to check for toxicity and determine safe dosages before any human trials. Next, consider how a double-blind trial reduces bias. The key here is that neither the patient nor the doctor knows who received the drug or the placebo, preventing both from reporting or interpreting changes incorrectly. Here is a quick glossary of terms to master. Remember, preclinical testing happens first. Toxicity measures harmfulness. Clinical trials involve humans. Placebos serve as control, and double-blind trials prevent bias from both sides. Drug testing also involves critical ethical considerations. While animal testing is controversial, it provides vital safety data. All clinical trials must ensure informed consent, meaning volunteers understand all risks and are free to withdraw at any time. Informed consent has three main components: providing full information about the trial and risks, ensuring participation is entirely voluntary with no pressure, and guaranteeing the volunteer's right to withdraw at any time without needing a reason. Why is randomization important? By assigning participants to groups by chance, we ensure both groups are similar. This prevents pre-existing differences, like age or health status, from skewing the results and making it impossible to evaluate the drug's true effect. This infographic summarizes the drug development stages. Remember that each phase is designed to build evidence of safety and effectiveness. Only drugs that successfully pass through every stage are approved for general patient use. Here are 4 must-know facts. Preclinical testing always comes first. Initial clinical trials use healthy volunteers. Placebos provide a control group for comparison. And double-blind trials keep both doctors and patients in the dark to ensure fairness. For top marks, use precise scientific terminology like toxicity and bias. Always explain the why behind testing methods, explicitly mention both parties in a double-blind trial, and link the reduction of bias to result reliability. You are ready for the exam. Remember, double-blind trials are superior because they eliminate bias from both patients and doctors. Good luck with your studies.